ASU iGEM is the undergraduate iGEM team from Arizona State University. For the 2022 competition cycle, we engineered the microalgal chassis Chlamydomonas reinhardtii to uptake arsenic from contaminated groundwater sources. This project was inspired by the widespread water contamination present in rural parts of Arizona as a result of decades-old mining efforts and a lack of water regulation. Our team's goal is to develop a solution that takes advantage of C. reinhardtii's natural metal-binding abilities to remediate these water sources in a way that's efficient and non-disruptive to the surrounding environment. To genetically engineer C. reinhardtii, we use the glass bead transformation method, which has been well documented as being successful in transforming genes into the C. reinhardtii nuclear genome. Multiple variations of this protocol exist in the literature, but the protocol below is a mixture of two protocols that gave our team many transformants. First, grow C. reinhardtii cell cultures in a baffled Erlenmeyer flask. We used a 125 ml flask containing 60 ml of tap medium. For 4 to 6 days prior to transformation, or until the cell count reaches around 3 × 10⁶ cells per mil. In our project, we used the strain CC400, which is a cell wall deficient strain of Chlamydomonas reinhardtii, and we suggest that if possible within the scope of your project, you also use a cell wall deficient Chlamydomonas reinhardtii strain to avoid the difficulties associated with transforming strains containing cell walls. 2. Transfer 30 to 50 ml of your cell cultures in tap into 50 ml falcon tubes and centrifuge them at 3500 G for 10 minutes. The resulting pellet should be easily visible and fully condensed to the bottom of the falcon tube. 3. After centrifugation, discard the resulting supernatant in a waste beaker. A dark green pellet of C. reinhardtii cells should remain at the bottom of the tube. 4. Resuspend the pellet in 1/100th volume of tap gently. If using 50 ml of cells, for example, resuspend in 500 microliters of tap, or 0.5 milliliters of tap. Avoid excessively agitating the cells by slowly pipetting up and down. 5. In pre-autoclaved glass tubes with 300 milligrams of sterilized glass beads of 425 to 600 micrometers in diameter, add the following in order: 2 micrograms of linearized plasmid DNA, your previously resuspended 500 microliters of cells, and 5% weight per volume of 20% PEG, polyethylene glycol. This is about 25 microliters if you're using 50 ml of cells. 6. Vortex the tube at the highest speed for 15 seconds immediately after adding polyethylene glycol. 7. Transfer the vortex cells into a 125 ml flask containing 30 ml of tap medium. Ensure that no glass beads are taken up alongside the transformed cells. 8. Place the flask on a shaker at room temperature for 24 hours in the dark to allow the cells to recover post-transformation. After the 24-hour cell recovery period, centrifuge the contents of the shaking flask down into a pellet at 3500 G for 10 minutes. 10. Discard the supernatant and resuspend the pellet in 1/100th volume of tap as you had used previously. Dispense the resuspension onto a selective tap agar plate and spread it evenly across the surface of the plate with larger plating beads. 11. Parafilm the plates to prevent desiccation and place them under direct light. Colonies should appear within 5 to 14 days post-transformation. Although glass bead transformation is a common transformation method for those working with C. reinhardtii, there are many factors that may be contributing to negative results. Firstly, it's important to ensure that enough cells are being used in the transformation. Our team transformed cultures of C. reinhardtii after an average of 5 to 6 days post-inoculation, or until the culture was a dark, rich green. Another unintentional factor that may reduce transformation efficiency is using circular DNA to transform rather than linearized DNA. Both approaches have the potential to result in transformants, but our team saw much success when using a linearized plasmid. Additionally, ensuring that the C. reinhardtii cells have adequate time to recover post-transformation is also incredibly important. Some protocols do not use a 24-hour dark recovery period, but our team had very little success in transformations that were plated without recovery.